Welcome to our T-cell series. T-cells are one of the most precise tools of the adaptive immunity, but they see the world differently than antibodies do. They usually do not recognize whole pathogens directly. Instead, they inspect small peptide fragments displayed on MHC molecules. So as we move through this series, keep one phrase in mind. Peptide plus MHC equals T-cell recognition. That simple rule will help us understand T-cell development, antigen presentation, CD4 and CD8 function, vaccine responses, and immune memory. Hi, I'm Christopher. In this video, we're going to take a big picture view of how T-cells recognize threats. Our goal is not to memorize every molecule involved in T-cell activation, not yet. The goal is to understand the basic logic of how T-cells see the world. And that world is a little strange. B cells and antibodies can recognize exposed structures on pathogens. T cells are different. They usually need antigen to be processed, chopped up into smaller peptide fragments, and displayed on another cell. That means that T cells are not just pathogen detectors, they are inspectors. They inspect molecular evidence, and based on what they see, they help decide whether the immune system should activate, help, kill, regulate, or remember. And that's why this topic is so foundational. So why does this matter clinically? Because T-cell biology sits beneath a lot of what we care about in veterinary medicine. When a dog controls a viral infection, T-cells are involved. When a cat responds to a vaccination and generates long-term protection, T-cells are involved. When a foal struggles with recurrent infections because antigen presentation is defective, T-cells are involved. When the immune system becomes too weak, too strong, or misdirected, T-cells are often somewhere in the story. T-cells help control viruses. They help coordinate responses to intracellular bacteria. They support antibody production. They activate macrophages. They help generate immune memory. Cytotoxic T-cells can also recognize and eliminate abnormal cells, including tumor cells. So this is not just immunology trivia. This is the logic behind infection control, vaccine success, immunodeficiency, hypersensitivity, autoimmunity, and clinical reasoning. If you understand how T cells recognize threats, many later immune system topics become much easier to organize. Before we zoom into T cells, let's place them within the adaptive immunity as a whole. Adaptive immunity has two broad arms, humoral immunity and cell-mediated immunity. Humoral immunity is all about B cells, plasma cells, and antibodies. This arm is especially important for extracellular threats. Think of bacteria outside of the cells, toxins, free viral particles, and other antigens that antibodies can physically bind to. Cell-mediated immunity is the T cell side of the story. This arm is especially important when the problem is associated within a host cell. That includes virus-infected cells, intracellular bacteria, and abnormal cells. But these arms are not enemies nor completely separate departments. They work together. For example, antibodies can neutralize free viral particles, but cytotoxic T cells are needed to kill the cells that are already infected. CD4 helper T cells support B cell antibody production, but they can also activate macrophages and coordinate broader immune responses. So rather than thinking humoral versus cell-mediated, think of adaptive immunity as a coordinated defense system. Different arms specialize in different parts of the threat. Here's the core rule for this video. T cells do not see whole pathogens. This is one of the biggest conceptual differences between T cell recognition and antibody recognition. A T cell is typically not recognizing an entire viral particle, an entire bacterium, or even a whole parasite. Instead, it recognizes small peptides displayed on an MHC molecule. That peptide is like a molecular clue, and the MHC molecule is like the display platform. The T cell receptor, or TCR, is the inspector that checks whether the peptide MHC combination matches what the T cell is built to recognize. That is why antigen presentation is so important. Without peptide display on MHC, most T cells would have nothing to inspect. No peptide MHC display means no meaningful T cell recognition. And that leads to one of the most important frameworks in immunology. The location of the antigen determines how it gets processed, how it gets displayed, and which T cells respond. A useful way to think about antigen presentation is showing ID. Cells display peptide evidence on their surface so T cells can inspect it. First, proteins are processed into peptide fragments. Those proteins may come from a pathogen, an infected cell, or material that an antigen-presenting cell has taken up. Second, those peptide fragments are loaded onto MHC molecules. Third, T cells inspect the peptide-MHC combination using their T cell receptor. If the T cell receptor, or TCR, recognizes the specific peptide in the context of the correct MHC molecule, that T cell can become activated. That is, assuming proper additional signals are present. The ID badge analogy is useful because the T cell is not just checking for any peptide. It is checking for the combination of the peptide and the MHC molecule presenting it. That is why we keep saying peptide plus MHC. The peptide tells the immune system what is being shown. The MHC molecule tells the immune system how it is being shown and helps determine what type of T cell will respond. This roadmap shows where we are headed. First, we will talk about the thymus. This is where developing T cells are educated. They must recognize self-MHC, but they must not strongly attack self-antigens. That balance produces T cells that are MHC restricted and self-tolerant. Second, we will talk about MHC. MHC molecules are the display system. MHC class 1 generally presents peptides from inside the cell and activates CD8 cytotoxic T cells. MHC class 2 generally presents peptides from material taken by the antigen-presenting cells and activates CD4 helper T cells. Third, we will talk about CD4 and CD8 T cells. CD4 T cells coordinate. They help B cells, activate macrophages, and shape immune responses. CD8 T cells kill infected or abnormal host cells. Fourth, we will talk about memory. After an immune response, most effector T cells die off, but some persist as memory T cells. Those memory cells help the immune system to respond faster the next time the same threat appears. So our story arc is this. T cells develop, antigens are displayed, specific T cells respond, and some of them remember. Let's bring this first video together. There are 5 big take-home points. First, T cells do not usually see whole pathogens directly. Second, T cells recognize peptides displayed on MHC molecules. Third, antigen presentation is how cells show molecular ID to the immune system. Fourth, CD4 and CD8 T cells have different jobs. CD4 T cells coordinate immune responses, while CD8 T cells kill infected or abnormal cells. Fifth, memory T cells help create faster future responses. So if you're only going to remember one pattern from this video, remember this. Peptide plus MHC leads to T cell response, and successful responses can lead to memory. That pattern will show up again and again in this module. In the next video, we will start at the beginning of the T cell story, the thymus. We will look at how T cells are selected, how they become MHC restricted, and how the immune system tries to prevent dangerous self-reactive T cells from escaping into the body. Thanks for watching. I'm Christopher Lee, and what you understand today helps you protect patients tomorrow. And I'll see you in the next video.